Excellent. Okay, everyone. So let's get started. So today we will talk about the basic molecular bonds and the interactions between the molecules in the living system. In fact, it also exists in a non-biological system. There are 5 types, so we will explain it one by one. And also we will get deeper into the 4 building blocks that we discussed, we overviewed in the last lecture. We will get to them in much deeper, and then we will highlight their functions in the life. Okay. Before that, for the announcement, there are just one quick announcement. So we will have midterm exam number 1 on September 29th. It will contain 10 questions. It contains questions that covers the material we will cover up to September 29th. Okay, and the exam will be on the class, so we will have 15 minutes. It should be sufficient to solve those 10 questions. Those 10 questions, some are asking you for the concepts, some are asking you to do very simple calculations, and some are asking you for some research type questions, thought experiment questions. Okay, so as long as you understand the material well, then these 10 questions will be like the practice for you. So it will be very fun for you to solve. Okay, and we are open book and open notes, so there should be nothing needed to memorize. All you need is to understand all the concepts and the materials we discussed in the class. Okay, so this is a call. Go ahead. Question.
Yes. Can you hear me?
I can hear you. Very good.
All right. I wanted to ask if any material on the midterm was going to come purely out of the reading and not based on anything we've discussed in class.
No. All the questions will be based on the slides and very little on the reading, because reading takes more time. So reading is more related to the homeworks. So then you do it after the class. Okay.
All right. Thank you.
Sure. For the exam, we target for the key concepts, important concepts, and especially to help you understand those concepts deeper. So then you are in a context of solving some questions, and then that question depends on your real understanding of the concepts. Okay. Good. Yeah. So have fun for the exam. Okay. Usually we let the students do exam for as long time as possible. As long as you like to work on those questions, then it's fine. Any other questions? But in our exam, I think 15 minutes should be sufficient. Yeah. Okay. Isabel have a question. Will there be any practice questions in addition to the homeworks? It's not necessary. As long as you understand the materials in the class, then you should be fine. Yeah. But we do. If you want, I can give you some example questions. That's also fine. Yeah. You hear me, Isabel? Yeah. Okay. Great. Okay. We have homework number 1 due September 25th, right? So you can work on that and submit it to me. Okay. And I will provide you with the solutions. Okay. So homework problem usually is more involved than the exam questions. Okay. But the exam question is more flexible. So the questions really test whether you understand the material or not. Okay. I think all of you are doing well. So just pay attention to read carefully the materials we discussed. Okay. For other updates, other announcements, they are the same as last time, and we will have more new readings provided to you, and also feel free to submit your survey to me, and let me know you have any opinions or any questions, any confusions in the class. Okay. Good. If no other question, let's start our today's class. So in last lecture, we started to talk about the building blocks of the life. and we discussed about there are 4 essential building blocks. There are 4 essential macromolecules in the living systems, and all the living systems consist of those 4 types of macromolecules. Okay, so first one here, there's no specific orders, so all 4 of them are important. The first one we discussed here is actually the DNA and RNA. They are made of, they are the polymers, and they are made of the nucleotide. The monomer is nucleotide, okay? ATGC nucleotide. For the RNA, they also have another different unit. Okay? Second one is protein. They are made of the monomer called amino acid. Okay? There are 20 types of amino acid. Okay? These 2 are they're kind of like the languages. They're kind of like they can compose sentences, paragraphs, and pages of the life. Okay, and the protein language is coming from the DNA and RNA language. Okay, third one is the lipid. They are made of fatty acid. Okay? They can form the membrane, cell membrane, that can protect organelle inside the cell and the whole cell. They can compose the whole membrane to protect the whole cell, to pack all the different organelles within the cell, to provide the protections. Okay? And also another important structural and energy components, building blocks, is called sugar. Okay, they are the branched carbohydrate. They usually made of the complex sugar are coming from the simple sugar. So they can have the condensation reactions so that they can polymerize and have the bigger complex sugar to be formed. And those carbohydrates can serve as the structure elements to maintain the cell shape. And also they can be used as the energy sources. They are not as flexible as ATP, but they can contain energy and then provide energy in a local region. They cannot travel for long distance like ATP does. Okay, so they can provide a local food for the cells. And we also discussed about the numbers of those 4 building blocks. They are they can be sub-categorized. And in total, the single monomer types building blocks for the nucleic acid, we have 8 types. Glycan, we have more than 32. The simple sugar also called saccharide. And then the complex sugar, we call it a polysaccharide. Okay? For the protein, we have 20 amino acids. And then lipid, we have about 8 types. But there might be more. There are more combinations. Okay, so here the types of molecular monomer building blocks are based on the chemical formula, and we have not considered the structure variation. We also talk about the isomer, right? Different structure variation and spatial change that also give different property of the building block. And then by definition, they actually also are different. They are also different alphabet. Okay. And then we did some example calculations. We saw how these 68 alphabets can be aligned together to form the sentences. And then by changing the alphabet, changing the geometry, they can have very complicated polymers. Even the alphabet only has 15 alphabet. They can have billions of variations, right? So that is the reason we have so many diverse creatures on the Earth. Okay, and those throughout the evolution, by using these 4 types of macromolecule, and then they are individual monomers. There are 68 monomers, but we using them, nature can help to construct, modify, and then allow the interactions between those components. Then cell can develop a really diverse shape and functions. Okay, and also here, all we are talking about is the genetic. Everything is coming from the genetic code. All those alphabet are coming from genetic code. Then they have those diversity. In the nature also there are non-genetic influence that can also change cell fate and cell behavior. That's called epigenetics. Okay, so that's why in the nature we have so diverse, things are so diverse. Okay? Now, all those monomers, if you look into those, you look into them deeper, you will find they are made of very basic chemical elements. If you look at those chemical elements, there are 6 very basic elements that are major part of those alphabets. Those 6 elements are hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur. These 6 are most common. And then these 6 elements, they actually have different valency, and then different electron negativity. Okay, the valency means they have different. If they remove the electron from those elements, they will contain positive charge there, right? And then those are called valency. Those valency electrons determines the bonding between the elements, and based on the valency and the weight, the proton of the element, they actually give they have the electronegativity properties. That property determines the polarity of the atom, of the element. Okay? If you look at in the living systems body, those 6 different elements, these 6 different elements, they have quite similar distribution. Okay, so here is one table. First they talk about these 6 basic elements. First 2 are the valency and the electronegativity of those 6 basic elements. Those are determined. Okay, but if you look at the human, third row and the 4th row, human and bacteria, in fact, the distribution of those elements, they are quite similar. The maximum, the distribution quite similar. Okay, that means the chemical composition of those living systems, actually they are not too dramatically different. Okay, and also the last row shows on the Earth what's for the non-living system, what's the distribution of those elements. Okay, so those are the elements, and are the most basic atoms. And then those elements actually combine together. They can due to those electronegativities, they can combine together, and then they can form bonds. And then those bonds together will give rise to the monomer we discussed about, amino acid, nucleic acid. Those monomer basic alphabets. Okay, and then that they can further form the bonds to get to the polymer, to get to the secondary structure and the tertiary structure. Okay, that gradually is a bottom up approach to form the living system. Okay, right now we are at the most bottom. This is the basic elements. Okay, so to understand how those bottom up avenue gradually give rise to the living system and then give rise to the function of the living system, we also needed to know how the bonds are formed between those elements and also between the different monomers. The people have already, the physics people, the physicists actually already studied the bonds properties between the elements. So here is a table for to summarize for the interactions and the pair potential, means the interacting energies between the two common atoms or the two common ions or two common small molecules in the vacuum. Okay, this is a non-living. So this is the purely physicists studied. And then they have measured the energy and the force between those atoms and ions and the small molecules. And they also listed the theoretical physics equation to calculate their bonding energy. Actually, they call it interaction energy. Okay, so those are really useful fundamental science. Give us the important sense what's the strengths between different the interactions. Okay, and this has been reported. This is actually a very good book. This book, the author is from UCSD San Diego. So they are the leading pioneer to study the intermolecular bond, intermolecular force between the atoms and also between the surface. Okay, so this is a very good book. Those interactions actually also happens in a living system. But in a living system, they're also the aqueous is always in the aqueous condition. So water solution always get involved. Okay, so the things actually become much more complicated in the living system. Okay? And in the living system, if we categorize those molecular and atomistic interactions, there are actually 5 types, different interactions. So we will go through it one by one. First, can you guys hear me? Can you guys hear me?
Yep.
Yeah, excellent, good. So all those 5 types of interactions are super important, and they are critical in different conditions, in different situations, and also in different scales. Means at a small nanometer scale or even to the micro and the millimeter or centimeter scale. Okay, in different scales. So we need to know which condition is most appropriate for those bonds to be used. So by understanding those 5 types of interactions, you can not only understand better how the cells interact with each other, but also you can do the reverse engineer. You can develop some engineering applications. This is called a by-inspired application. Okay, so you can develop some tools that mimic the nature and provide a new utility to the human being. Okay, so that's why it's very important. First type of interaction is when the molecule, when the atom form the covalent bond. So covalent bond actually is when the 2 atoms are held together by sharing an electron. So on the surface of the atom there are electrons, right? So if you look at the picture on the right hand side, this is 2 hydrogen atoms. They are close together. For the hydrogen bond, the orbit surrounding it, the most basic orbit can only hold 2 electrons. So when 2 hydrogen atoms are close by, they actually can attract each other, and then they can take the electron of the neighbor's atom and then share them together. Okay, so they share together. So on each atom's the orbit, they have 2 electrons. One is from their own, another is donated by the neighbors, shared by the neighbors. Okay, and then this shared 2 electron actually stay in the relatively constant position. They cannot rotate anymore. They are attracted and form a dipole here. Okay, they stay relatively stable here. This actually is a single bond. It's a single bond. Okay, if there are multiple electrons here, then you can form multiple bond, such as the double bond that mostly existing in the carbon-carbon atoms. And then rarely they can have a triple bond. Okay, so those bond, those single bond, double bond, and triple bond, they are formed by sharing the electron, by the collaborations between different atoms. This condition, this kind of bond is called a covalent bond. Okay, you may already learn those previously, but right now let's get it deeper. This bond actually is very strong. When the people form, when the atoms form this kind of collaboration, the bond, they are very, it's very hard for them to be separated. Okay, once they are married, they don't divide. Okay, and then those bond actually, the bond force is about 1 to 10 nanonewton per bond. Each bond has 1 to 10 nanonewton. That's actually pretty strong. Distance is about 1/10 of nanometer, 0.1 nanometer. Okay, and as I said, the electron, once they adhere, once they attract each other and form the single bond, they actually stay in the position quite stably. So that means the bond, direct bond, this covalent bond has a pretty clear directionality. Their direction is from the neutron, from the nuclear 1 to the nuclear 2. So this is the horizontal way. Okay, so it's very directionally. They cannot move random. So this is one direction. And the bond angle is determined by the unshared electron pairs. So if there are other unshared electron pair for the even higher order atoms, then they will determine the other electron, if they exist, they can adhere or repel this two pair of electron, so that it can vary the angle a little bit. But once they are formed, the angle will not change too much. Okay. So to understand those covalent bond, and as well as future other bonds, there are several properties you need to understand. Firstly, what is the nature of this bond? Means how this bond is formed, how this interaction is formed. Here, for the covalent bond, they are formed by sharing the electron. Second one is what's the bond strength? What's the force needed to break that bond? Or sometimes people use energy. What's the potential energy that formed in this bond? And if we break this bond, what kind of energy will release? That is equivalent energy. Okay. And the third one is what's the distance between those bonds? Number 4 is whether those bond has directionality or not. Okay. So this actually is very useful. Some in the engineering applications, sometimes you need the bond to have directionality if you want to have the final product to be stable. But some other times you want the bond to be non-directionality. Then you gain more flexibility. If you don't care about the bond, the structure to be stable or not, you want to have a flexibility. Then you need to choose. You need to avoid covalent bond. You need to choose other bond that has no directionality. Okay. So those are the key properties you need to know. Second important interaction is actually from the ionic bond. Ionic bond. So ionic bond actually is formed by two atoms that hold together by the electrostatics. That actually always happen when two atoms that have very dramatic different electronegativity. One kind of atom or one kind of element is very strong in attracting the electron. Another one is relatively weak. For the covalent bond, actually they are quite equivalent. They cannot defeat each other for two atoms. But for the ionic bond, the two atoms, one can very easily, very obviously defeat another one and then grab the electron for the of that guy. Once they grab that electron, they become very negative. And then the one being stripped of the electron become positive. Then they can attract each other, form a bond that is called ionic bond. Okay. So one example is sodium chloride. So for the chloride, it's very, very strong in grabbing the electron. Okay, so then the orbit of chloride become satisfied, and then the sodium is actually very easy to release to give up their electron. Okay, so then the chloride become negative, sodium chloride ion become negative, sodium ion become positive, and then they attract each other. In this case, in fact, the two electrons on the surface, they act on the surface of chloride, they can go quite freely. They can go freely, can rotate freely. Okay, that actually makes this sodium positive ion can also move around the chloride surface. So that means the bond, the ionic bond between these two does not have a directionality. They can go different directions. They don't have different. They don't have a persistent direction. Okay, so this is a non-directional bond. The bond actually is also very strong. It's about 100 piconewton. Okay, 100 piconewton per bond. And the distance, but the distance can be larger than the covalent bond. The bond is weaker than the covalent bond, but the distance can be larger, 0.25 nanometer. Okay, and the ionic bond is no directional because the ion has the spherical symmetry of their electric field, because those electrons can be equally distributed in anywhere on the surface, on the orbit of the chloride ions. Okay, so these two actually are very useful bonds. They for the first one, they actually give rise to the backbone of the polymer. For the second one, they actually very useful in the solutions, in the solutions. Okay, and then those solutions give rise to the activation of the proteins, and also can transform the protein, can make the protein to be phosphorated, or make the protein to change their properties. Okay, that's epigenetic effects. Third one is called a hydrogen bond. So that usually is coming from the electrostatic dipole interactions between the water molecules. Okay, so in this case, on the right hand side, you can see the two water molecules. The water has oxygen and hydrogen, one oxygen atom and two hydrogen atoms. Another one also has the same formula, but because the oxygen and hydrogen have different electronegativity, so water molecule actually it actually has a distribution of charge on its surface. Close to the oxygen, they are more negative. Close to the hydrogen, they are more positive, because the electron donated by the hydrogen is more close to the oxygen. Between the oxygen and hydrogen, this is a covalent bond. They actually share two electrons. But because the water molecule has this charge distribution, positive and negative charge distribution, then between water molecules, the positive part closing to the hydrogen actually can attract the negative part from the oxygen atom of a neighboring water molecule. Then they actually can adhere to each other. That forms a kind of interaction bond called a hydrogen bond. This happens a lot. This actually happens in the proteins. In the protein, there are a lot of hydrogen and oxygen. As we show, there are 6 types of different basic chemical elements, right? Oxygen and hydrogen are inside. And then for the amino acid and the proteins, they actually have a lot of hydrogen and oxygen inside. And then they also form the charge polarity. And then those charge polarity can adhere each other to bend the amino acids chain. That forms the shape, 3D shape of the proteins. Okay, so in the protein, they actually have 2 types of basic subunit. One is called alpha helix. So this is a helix, alpha helix. They form this helix is because between each rotating region, there are many hydrogen and oxygen bond atoms. They form the hydrogen bond to adhere each other. So then they can bend the polymer into the alpha helix. Second type is called beta sheets. Those beta sheets actually is quite straight. It's like a sheet. However, between different sheets, they can actually go in parallel. You can see those blue colored sheets. They can adhere to each other in parallel, because between the sheets there are hydrogen bonds. Okay, so those are very fundamental bonds to form to constitute the larger shaped molecules, to form those macromolecules. Okay, for the hydrogen bond, the bond width actually relatively small, is 10 piconewton, much smaller than the ionic bond, 10 piconewton per bond, and the distance actually is larger, is about 0.3 nanometer. And as we said, they formed because of the negative and positive attraction, right, of the specific region of the water molecule. So then their direction is very determined. So the hydrogen bond has directionality. Okay. So this is the third type of interactions. Up to now, you can appreciate why those interactions are important. They actually generate different kind of functions and the structures of basic atoms. Right? They gradually build up the things into more interesting functions. So for the water, it is polar because it's slightly negative. As I said, negative oxygen and slightly positive hydrogen. Then they can form the hydrogen bond. Also in the DNA, they have the different base pair, and then for the ATGC, A is to the T, G to the C, right? So it's 1-to-1 base pair. This base pair pairing is also due to the hydrogen bond. In the protein structure, as I just mentioned, they can form the alpha helix, beta sheets, and they can actually, those can go even higher order. They can have many of them combined together to be larger proteins, larger size macroproteins. Okay, they are all formed by the hydrogen bond. Number four is called number 4 interaction is called van der Waals interaction. Okay, so this kind of interaction actually is a very flexible van der Waals interaction. It is driven by the induced electrical interactions. So for induced interaction, so it means for the any molecule, for example, we have two molecules, two single or two atoms. Let's talk about the molecular scale. We have two single atoms here. So they contain so these two not necessary in the water. They contain positive proton inside and a negative electron outside surrounding it, both of them. If due to some environment influences, those atoms show some electrical density change. For example, for this atom, if they are electron, for some reason, they all go to the left-hand side or right-hand side of it. So they form this kind of distribution. Then this atom overall is still neutral. But if you look into its distribution, they actually is positive on the right-hand side, on the left-hand side, and then negative on the right-hand side, right? So they have a polarity within itself, although overall is neutral. And then for the second atom, if they show the same kind of distribution, then the left-hand side positive region of the second atom can attract the negative side of the first atom on the right. Then they actually can attract each other. When they attract each other, they actually form a very stable interaction. And then if these two atoms continuously show this kind of electron distribution, then this bond it persists there. Whenever persists as long as electrical distribution persists. Okay, this kind of bond is called a van der Waals bond. We call a van der Waals interaction. So this is driven by induced electron interaction. So there's a transient shift due to the environmental influence. Okay, the transient shift in the electrical density between two or more atoms or molecules that are very close to each other, right? So the close means they actually have the distance is 0.3 to 0.6 nanometer. Okay, and then this attraction, because this is due to the electrical attraction between two atoms, they are actually the distance dependent. If the distance is too far away, distance between the two atoms too far away, then the electrical attraction is very little. Remember the Coulomb theory in the electricity. There, the R square is at the denominator, right? Q times Q divided by R square. So if the distance is too far away, then it's a square root, is square. So then the attraction is very, very small. But if they are very close, if R is very, very small in the denominator, very, very small, then the Coulomb attraction is very large. The force is larger. Okay, and then this atom actually can change its direction. It can rotate, and then the van der Waals force is still existing there. Okay, so this van der Waals force also has no directionality. It's not like a covalent bond. It also has no directionality. Okay, and it's also different from hydrogen bond. No direction, so they can rotate. And the bond strength, so this strength is even smaller than the hydrogen bond. So it's a 0.1, 100 times smaller than the hydrogen bond. It's a 0.1 piconewton per bond. Okay. So this van der Waals force actually is very transient. It's very transient. It's depending on the distribution of the electron around the atoms. So this actually has a great advantage. The property I just mentioned here has great advantage that nature already leveraged for millions to billions of years. One example is the glycol. So on the right-hand side, there's a picture showing the glycol. So we all know. Do you guys hear me? Do you hear me?
Yes.
Yes. Yeah. Okay. Great. We all know glycol can work on the wall very rapidly, right? And then they can capture the mosquito, flies very, very quick on the wall, right? Why glycol can adhere to the wall? The reason is glycol is taking advantage of van der Waals interaction. It is not using the hydrogen bond. The hydrogen bond is mostly in the water. Okay, and then it cannot use the bond like in the covalent bond or the ionic bond. They are too strong. The glycol, in order for glycol to move quickly, so here are two features actually. Let me explain one by one. Two features for the glycol to work to run on the vertical wall, or sometimes even actually it can work underneath the roof, right? So the feet of the glycol is it has a very strong interaction with the roof, and then that interaction sustains the whole weight of the glycol, right? So for glycol to have this capability, there are actually two features. One is why this small feet, very small feet, can form strong interaction with the roof or with the wall to sustain the weight of the glycol. Okay, the means a strong bond. Why they have a very strong bond there? Second is why those glycol can run very fast on the roof. Okay, that if they can run very fast on the roof, that means the bond between the feet of glycol to the roof actually can form and break very fast. If they can form and they can form it, but they cannot break, then the gecko's feet is sticked on the wall. That is not good. They cannot capture the mosquito and flies. They can notice the danger very, very quickly, and then they can flee, right? Then gecko will not be able to capture the food, right? So nature designed in a way that a gecko can form the strong bond on the feet, and also that bond can disassemble or assemble in a very quick way. In fact, people have categorized the gecko's feet. They find the bond, the van der Waals force between the feet and the roof can turn on and turn off in 10 milliseconds. 10 milliseconds. Okay. And what's the reason they can have such a strong bond? The reason is on the feet of gecko. If you use the microscope, zoom in to the feet, you will find there are many fibrous structure. Like here, you see the fibrous structure. This is all in a micrometer to nanometer scale. So those fibrous structure actually is very small. It enable, because they are very small, they enable the gecko to apply force to the wall, and then let this filament to get very close to the wall surface, and then that force applied can change the electron distribution on the surface of this fiber, this fibrin structure. Once this electron distribution is changed on the surface, that can adhere to the wall, form this electrical attraction here. Okay, form this attraction, and there are many of these fibers. So there are many of the small force added together. They all added together to have a force summary. That summary actually can let the gecko to sustain the weight. Okay. Based on this, in fact, there are several things we can appreciate. One is those fiber on the feet actually should be as small as possible. If they are very small, then also, and we have a large number of those fibers. By being smaller, those fibers can get closer to the wall, and then the electrical distribution on the fiber can be easier to be perturbed by applying the force, okay, through the gecko's muscle. And being denser, many of them, then we have many small force can be manipulated by the gecko, by the force. Okay, then they together can enable the sustaining the weight of the gecko. Okay, and here also I mentioned about the speed, right? So about the speed, the reason the gecko can quickly disassemble those filaments is because they are very smart to change the orientation of this filament. When the gecko need a big force, they actually push those filaments down to the wall or to the ceiling, roof, so that the longitudinal length of those filaments are adhered to the wall. So they actually have more contact area. So he uses the longitudinal extension of the filament contact the wall. So then they have sufficient part of filament to form a van der Waals interaction with the wall. When the gecko want to capture mosquitoes, then he actually changed the direction, changed the orientation of the filament. They only use the tip, so change it 90 degrees. So only the tip of the filament contact with the wall. So then it's only the cross-section area contacted with the wall. Then the force is much smaller. That force can be easily ruptured from the wall. And then they go to the next position. And then at next position, they rotate 90 degrees again to let the filament flat down and then adhere to the wall. Okay. And then gecko has 4 feet, right? So when they do it, they actually, the opposite side Do you guys see my mouse? See my mouse? Do you guys hear me?
Yes.
Yeah.
I can hear you.
Okay. When the glycol runs, actually they use the opposite side, release first, and then like this one and this one release, and then these two still adhere, and then they make the leap forward, and then the previous detached two adhere again, and then the next two release. Okay, change the rotation, change the angle of the fibrin. Okay, so that's how the glycol leverage the van der Waals interaction. Okay? And in the nature, actually, people find there is another scaling law here. Another scaling law. We know all those beetles, flies, and spiders and the glycol, they all are able to adhere to the surface of the roof, right? They all can do. But but their weight is different. From the beetles to fly to the spider to the glycol, the weight increase. And then as the weight increase, if you look at the deeper into the van der Waals interaction of those animals to the surface, you will find the functional unit that give rise to this van der Waals interaction. Those fibrillary structure actually get more and more tiny. For the glycol, they become very, very tiny. Okay, there is a scaling law, very tiny, and they have many of those filaments. Okay, and if you counter the density of those filaments, they actually proportionally increase as the mass or the weight of the animal increase. Means they have more and more. This actually has more and more contact area. They have more contact area to allow bigger weight to be sustained through the van der Waals interaction. Okay, so the density of the surface area actually scale with the body weight. This is another scaling law existing in the nature. Okay, so that's why those molecular interactions are useful. People actually are using those engineering the devices that the human being can carry to use this van der Waals interaction so that we can climb on the building. Okay? Without using any glue, we can easily climb on the building. Yeah. So that will be useful for the application in the extreme environments. Right? Number 5 of interactions called hydrophobic interaction. So this interaction actually is because the molecules contains both the hydrophobic region and hydrophilic region. Hydrophobic means the molecule heat the water, such as the lipid, they heat the water. Hydrophilic means they actually like the water. They usually in the physical chemistry, people use the contact angle to characterize the hydrophobicity. Okay? If the angle is very large, means like the water droplet on the lotus leaves, right? So that is hydrophobic. And if the angle is very small, then it's a hydrophilic. And in the liquid, if we have a larger macro molecule, it contains hydrophobic region and hydrophilic region, then to minimize the free energy, they actually can self-assemble. Hydrophobic and hydrophobic part can get aggregated together, and then they tend to stay inside of the macromolecule to let the outside region to be occupied by the hydrophobic region. Okay, so eventually they will form this kind of shape in the water solution. Okay, this interaction, this kind of assembling process is driven by the hydrophobic interaction. So this definition is it is the aggregation of nonpolar. Usually nonpolar means hydrophobic. Okay, because water molecule is polar. So nonpolar molecule in the aqueous solution, and then maximize the hydrogen bond between the water molecule by excluding the water molecule. Okay, so that's the magnetic explanation of why the hydrophobic region will will tend to stay in the middle. Okay? This fifth interaction actually is non-specific. This is formed by the self-assembly. This is non-specific. As long as they non-specific means they are not specific to the element types. They are specific to the. They are only depending on the hydrophobicity, okay, or hydrophilicity. Okay, so when this kind of structure is formed, there are no directionality. Okay, and this behavior actually many scientists believe at the in billions of years ago, when the human being when there is no human, when there is when the central dogma started to exist from the DNA to the RNA and RNA to protein, when central dogma started to work, they firstly use this hydrophobic interaction to let the DNA, in fact, first is RNA. They first let the RNA gradually to self-form this shape, those kind of complex shape, and then the proteins come in to reverse the RNA into the DNA, because DNA is more stable. Okay, so that very initial the form to let the life started actually is due to the hydrophobic interactions. Okay. And this interaction, this hydrophobic interaction also existing in the cells for another very important phenomenon called the phase separation in the cell. So we talked about previously, we talked about the compartmentalization of different organelle in the cell, right? So those different organelle separate from each other because they have a membrane on the surface. But even without a membrane, those macromolecules, they can also self-assemble into this kind of structure, and then they will aggregate together without a membrane. Okay, so here is one example of the cells that shows a membraneless organelle. So here, those organelle actually coming from those aggregate of this, or many of those same types of proteins, and then they form those yellow dots, yellow aggregates. Those are all non-membrane enclosed organelle, but they actually function their function are very important. Cell actually has a certain unknown capability to instruct the formation of those new organelle. And then those new organelle, the advantage is it can locally increase the concentration of certain protein, and then to speed up the chemical reaction. Yeah? And then once the chemical reaction is done, the function is realized, cell can disassemble those organelle. So those organelle is not permanent, they're transient. And then they can disassemble them. And then cell can instruct other types of membraneless organelle and then do other function. So this is actually a new field. People study the phase separation in the cell. So you look at this, it's like the oil in the water, right? So each membraneless organelle is like an oil droplet in the water, right? So those are the localized. There are many localized reactions happening inside, and those are all due to the hydrophobic interactions. So this is super important. All 5 types of interactions are very important. It's just depending on which scale, which spatial scale, what kind of time domain you are interested in, and what kind of function that you are studying. Okay? And those we need to understand all these 5 types. Yeah? Okay, now it's 3:54. We can continue our discussion on the Wednesday. Yeah, very good, very good. Do you guys hear me?